closer look at memory, the effect on diurnal rhythms on perineuronal nets.
Hi everyone. I'd actually like to start with a question. Let's do a show of hands. How many of you feel like you don't get enough sleep? Exactly, me too. We all know that a good night's sleep is important, but not only is sleep important for our physical bodies, it's extremely important for our mental function as well, especially with regards to memory consolidation, the process of converting short-term to long-term memory. And even though we spend a third of our lives asleep, we still know very little about what actually goes on in the brain when we go to sleep at night. While we usually consider how neurons influence our brain function, it turns out that the material surrounding our neurons, the perineuronal net, or the extracellular matrix, plays an extremely important role as well. In the Sorg lab at Washington State University, we focused on one particular brain structure, the perineural net, shown here in green. And these perineural nets have been shown to have a relationship with memory consolidation and regulating neuroplasticity, which is why we want to investigate them. Ultimately, our basic research question was, how do these natural sleep-wake cycles affect these perineural nets? And from this, what can we gain about the relationship between sleep and memory consolidation? In order to conduct our experiment, we used a rat model. And although rats are nocturnal, they actually provided an excellent model of the human brain for us to study. To see the changes in the intensity and activity of these perineural nets over a 24-hour sleep-wake cycle, we looked at these structures at 4 different time points within a 24-hour cycle. Each of our brain slices was stained with different fluorescent proteins and then magnified and imaged using a confocal microscope. And an example of that image is shown here. We then used these images and applied them to a unique pipsqueak software that we developed in the Sorg lab. This software allowed us to analyze and measure the intensity of these perineural nets and neurons 100 times faster than doing so manually. And after going through a lot of difficulty with troubleshooting this software, that was how we were able to gather our data. So what did we actually find? Essentially, our take-home finding was that these perineural nets actually became thinner during sleep. Essentially, as these nets became thinner during sleep, more so-called holes could appear in the net, and we believe this could allow for more synaptic connection to occur with the neuron. And this increased synaptic connection, we believe, could potentially lead to more memory consolidation. And that was our most important finding. So what's next for us at the lab? We believe that this research provides an excellent baseline for us to continue studying more research involved with sleep and perineural nets. We hope to look into how sleep deprivation, oxidative stress, and a variety of other factors influence perineural nets. And with a better understanding of these nets, we hope to get closer to understanding the relationship between sleep and memory. And at the Sorg lab, we are glad that we are one step closer to the answer. Thank you.